Namaste children, welcome to biology class. Today you will learn about the interesting topic is transpiration. What is meant by transpiration? The loss of water in the form of water vapor from the aerial parts is called transpiration. The transpiration can be classified into three types: stomatal transpiration, cuticular transpiration, lenticular transpiration. Now watch the video.
Transpiration, Part 1. The sun heats up the water in puddles, rivers, lakes, and even oceans. As water gets warmer, it begins to evaporate into the air as water vapor. Trees and plants too give off water vapor from their leaves through the process known as transpiration. The loss of water in the form of vapor from the aerial parts of plants is known as transpiration. It is one of the most vital phenomena in plants. Transpiration can be easily demonstrated in a laboratory. Take a well-watered potted plant. Cover the soil surface properly with a sheet of oil paper. Then place the plant in a bell jar and keep it as it is for some time. Observe the bell jar. You will notice water droplets on its inner surface. This water has come from the aerial parts of the plant due to transpiration. Three types of transpiration have been recognized: cuticular transpiration, lenticular transpiration, and stomatal transpiration. Loss of water from the surfaces of epidermal cells of leaves and herbaceous stems is said to be cuticular transpiration. Since surfaces of epidermal cells are often covered with a cutin layer called the cuticle, such a transpiration is called cuticular transpiration. Cuticular transpiration accounts for up to 10% of the total water loss by the plant. Water loss through lenticels present in woody stems is called lenticular transpiration. Due to the limited distribution of lenticels, lenticular transpiration accounts for less than 1% of the total water loss by the plant. Loss of water vapor through the stomata is known as stomatal transpiration. About 80 to 90% of the total transpiration by the plant occurs through the stomata. Stomata are minute pores in the epidermal layer of the aerial parts of plants. They are most abundant on leaves. A stoma is surrounded by two specialized epidermal cells called guard cells. The guard cells are usually kidney or bean-shaped with thick inner walls. Unlike other epidermal cells, guard cells contain chloroplasts and carry on photosynthesis. The size and shape of the stomatal aperture varies with the turgidity and shape of the guard cells. The number of stomata ranges from a few thousand to over 100,000 per centimeter square area of the leaf, and they occupy 1 to 3% of the total leaf area. When the stomata are open, there is an easy diffusion of water. The mesophyll cells lining the substomatal cavity are in direct contact with the air, and water is lost from their surfaces. Based on the time of the stomatal opening, stomatal movement is of two types: photoactive movement and scotoactive movement. The photoactive movement is controlled directly or indirectly by light, while the scotoactive movement is not controlled by light. In photoactive plants, the stomata open by day, whereas in scotoactive plants, they open at night. Only a few plants like cam plants show scotoactive stomatal movements. Stomatal transpiration occurs only when the stomata are fully open. It increases rapidly with the increase in the size of the stomatal aperture. The opening of stomata is governed by the changes in the turgor of the guard cells and the epidermal cells adjacent to the guard cells. Osmotic changes in the guard cells cause water to move in or out of the guard cells. When water moves in, the guard cells become turgid and the stoma opens, and when water moves out of the cell, it becomes flaccid, which results in the closing of the stoma. Due to an increase in the osmotic contents, that is, the sugar content of the guard cells, a diffusion potential deficit, or DPD, gradient develops between the guard cells and the neighboring cells. This causes diffusion of water into the guard cells, causing them to expand and become more turgid. A decrease in the osmotic contents of the guard cells develops a DPD gradient in the opposite direction. This causes the diffusion of water out of the guard cells into the neighboring cells, making the guard cells flaccid. There are two main theories put forward to explain the changes in the osmotic pressure of the guard cells: starch-sugar interconversion theory and active potassium ion transport mechanism. Let's take a look at starch-sugar interconversion theory. The stomata open in light and close in darkness. This is due to enzymatic conversions of starch into sugar in light. The soluble sugar, which is glucose 1-phosphate, is formed as starch plus inorganic phosphates in the presence of light produce glucose 1-phosphate, while in darkness, in the presence of phosphorylase enzyme, glucose 1-phosphate is converted back into starch and inorganic phosphate. The conversion of starch into soluble sugars increases the osmotic concentration in the guard cells. As a result, guard cells absorb more water from the neighboring mesophyll cells. The turgor of the guard cells thus increases, resulting into the opening of the stomata. In the dark, sugar is transformed into starch, and the whole process is reversed, leading to the closure of the stomata. Sayeri, in 1926, further elaborated this hypothesis. According to him, a change in pH affects the opening and closing of the stomata. Light increases the hydrogen ion concentration and darkness lowers it. When pH is high, the enzymatic conversion of starch to sugar is favored, and at a low pH, there is a reverse process and starch synthesis takes place. It has also been suggested that the increased pH may be due to the consumption of CO2 in photosynthesis. Scarth, in 1932, supported Sayeri's view. He explained that CO2, released by the process of respiration, is exhausted in the process of photosynthesis by day, raising the
This is a structure of stomata. This is the stomata surrounded by guard cell. This is stomatal pore. This is nucleus.
pH value of guard cells. High pH favors opening of the stomata. At night, CO2 released by respiration is not utilized in photosynthesis and thus gets accumulated and dissolved in the guard cell sap, forming carbonic acid, resulting in a drop in the pH. Low pH favors closing of the stomata. Steward, in 1964, considered that ATP is essential for providing energy, for opening and closing of the stomata. According to him, glucose 1-phosphate cannot chain osmotic pressure to such an extent which can induce the stomata to open. According to Steward, in light, glucose 1-phosphate is further converted into glucose 6-phosphate and then into glucose and inorganic phosphate. The glucose and phosphates get dissolved in the cell sap and lead to the enhancement of osmotic pressure, thereby DPD of the guard cells. Water is then influxed into the guard cells from nearby cells. Guard cells become turgid, causing opening of the stomata. At night, photosynthesis stops and the level of CO2 is high in substomatal cavity, which results in the lowering of pH of guard cells. Glucose molecules are then converted into glucose 1-phosphate by utilizing respiratory ATP. The glucose 1-phosphate is then converted into starch.
This tree, which is several meters tall, need water. Plants are not mobile. How do they manage to get water right up to the tallest branch? The roots of plants absorb water and mineral salts from the soil through the root hairs by the process of osmosis. Only a small percentage of this water is used by the plant. The rest gets evaporated from the surface of leaves. This evaporation of water from the plant is called transpiration. Let us perform a simple experiment to show that transpiration takes place in plants. Take a medium-sized, well-watered potted plant. Cover it with a transparent polythene bag A and tie its mouth at the base of the stem. Next to the potted plant, place an empty polythene bag B with its mouth tied. Expose this setup to sunlight for an hour or two. Drops of water will appear on the inner side of the bag A. The polythene bag B will not show any water drops in it. The leaves give off water vapor, and due to condensation of the water vapor, tiny drops of water get collected. This experiment shows that plants give out water to the atmosphere through transpiration.
I included this video for your better understanding. Okay, children?
There are three types of transpiration depending on the site of transpiration. They are stomatal transpiration, lenticular transpiration, and cuticular transpiration. Stomatal transpiration. Stomata are openings on the leaf surface through which transpiration takes place. The stomata are surrounded by guard cells which help the stomata to open and close. Lenticular transpiration. Lenticels are minute openings on the stem, and a small amount of water is evaporated through them. Lenticels are less in number compared to the stomata. Cuticular transpiration. This is the transpiration which takes place from the leaf surface. Leaves are covered by a waxy layer of cuticle. The cuticle actually helps the leaf to reduce the amount of transpiration taking place. Water is absorbed by the root of a plant. The tiny root hairs help in absorption. The water is then conducted to the upper parts of the plant through the xylem. The xylem cells have thick walls and can be compared to the pipes carrying water from the underground tank to all parts of the house. The mesophil cells in the leaf have large intercellular spaces between them. The evaporated water vapor collects here and goes out through the stomata, which open and close periodically. Water is also evaporated from the surface of the leaf through the cuticle. When the water from the outermost layer gets evaporated, it absorbs water from the layer of cells below it, and this process creates a pull by which water gets pulled up through the xylem. Transpiration is very important to plants, as the transpiration pull helps in water rising to the leaves against the force of gravity. This activity will show us that both surfaces of the leaf do not transpire at the same rate. We use cobalt chloride paper to test for the presence of water. Cobalt chloride paper, which is normally blue, turns pink when it comes in contact with water. Take 2 strips of cobalt chloride paper and place them on either surface of one of the leaves of a potted plant. Cover them with a glass slide and hold them in place with elastic bands. Leave it for an hour. Now remove the strips and observe them closely. What do you see? The strip placed on the lower surface turns more pink than that on the upper surface. This shows that transpiration is more on the undersurface than on the upper surface. Do you know the reason for this? The upper surface gets more sunlight and the plant would lose most of its water if the number of stomata on both the surfaces were the same. Though transpiration is essential for plants, they also adapt themselves to minimize the water loss through transpiration.
Now let us see the significance of transpiration. Here firstly it creates a pull in leaf under stem. It regulates the temperature of the plant. It is necessary for the continuous supply of minerals and water. These are significance of transpiration. Okay children, it was interesting this video. Yes, I hope you have understood well. Thank you children. See you next class. Another one topic. Thank you. Bye.